So it's my great pleasure today to introduce Carsten Allefeld from the City University in London. And he will be joined also by some parts of the talk by Thomas Christophel here from Berlin. We are very happy that you are here and are introducing us to some advanced methods in the field of decoding from fMRI and other brain signals with some advanced methods beyond classification. Carsten was doing his PhD at the University in Potsdam in 2004. I see from your CV, then you were around a bit, and then the time where most of you will know you from is that you were an associate at the Bernstein Center for Computational Neuroscience here in Berlin from 2009 to December 2018. And then Berlin lost you, unfortunately, to London. As you were saying earlier, Berlin's the best city in the world, and now London is also okay. And you moved to London, and since then you are a lecturer at the City University in London. And keep going with interacting with Berlin and developing further some methods that then we are happy to use also in our research. And we are happy to learn from you about some some advances in the multivariate classification techniques that are now on the market. So thank you very much for being here, and stage is yours.
Thank you. I was wondering, maybe we should allow questions in between, or do you rather have them all at the end?
I think both is fine. Yeah.
Okay. So then just please interrupt me if something's unclear. Good. Okay. Now to find out how to share the screen. Okay. Can you all see that? Oops.
Yes, very well.
Yeah. Okay, good. Okay, so I'm going to talk about multi-voxel pattern analysis. The word voxel already implies that it's mostly geared at fMRI, but I think it can also be applied to EEG, even though we haven't explicitly tried that. Multi-voxel pattern analysis by cross-validated MANOVA and by cross-validated cross-MANOVA. So the first part of the talk is a recapitulation of what we have done before. That was work at the BCCN with John Dylan Haynes. That was a cross-validated MANOVA paper, I think 2014. And then in the recent years, Thomas and I, and also a PhD student of his, Polina Yamchichina, we talked about how we could do in cross-validated MANOVA what is done in cross-decoding. So cross-validated MANOVA is a form of decoding, if you will. And so there will be a second part about extending cross-validated MANOVA to the cross situation, the cross-decoding situation, which we are calling cross-MANOVA. First, a brief introduction into the strategy, decoding versus modeling. I assume that most of you are familiar with the idea of multi-voxel pattern analysis. The idea is to assess information content of patterns of activations, where these patterns are across multiple voxels, as opposed to traditional mass univariate fMRI analysis. So you get a number of voxels. Those can be the voxels in the searchlight. The searchlights move throughout the brain, or they can be the voxels from a region of interest, or if you want, they can also be all the voxels in the brain. And you put them together into activation vectors. So you have fMRI data or beta values from a GLM in the different vectors, and the vectors go across different voxels. The vectors are as high dimensional as the number of voxels from which you construct them. And then you look at these activation patterns in different conditions, for example, here condition A and condition B. And you imagine these vectors to describe points in a high dimensional space, which is visualized here as a two-dimensional space spanned by two voxels. And the idea is that in this high dimensional voxel activation space, different experimental conditions may be better distinguishable than they are in each voxel separately. So that's the idea of multivariate analysis or multi-voxel analysis. Now, the traditional way to do this, or the standard way to do this, is to use so-called decoding or classification. So again, you have this voxel activation space symbolized by two voxels, voxel 1, voxel 2. You have these points which belong to different conditions, A and B, green and magenta something. And then you train a classifier on part of the data, which is indicated by this green, magenta dividing line. And you see how many of the remaining part of the data, which you haven't used for training this classifier, lie on the correct side. Then you can quantify the performance of this classifier by an accuracy. In this case, for example, 75% of the unseen data points were classified correctly. And then this number, this accuracy, is a measure of the information content, the multi-voxel information content. Now, there are a number of disadvantages to this decoding or classification approach. The accuracy is an indirect measure, it does not describe the data, so you can't say, okay, this is in the data what accuracy describes. It describes the data in combination with the classifier. The accuracy depends on which level you apply classification. You can apply it to single volumes, or you can apply it to GLM parameter estimates on the runwise level. It depends on the partitioning of the data, so how much of the data is used for training, how much for testing. It may depend on the properties of the specific classification algorithm. If you have a small number of values, for example, if you do classification of runwise parameter estimates, then you have a limited range of discrete values. So the accuracy can be 50% or 55% or 60%, but nothing in between. And finally, classification is only defined for patterns which belong to discrete classes. Now, an alternative to that would be to use a generative model to describe the data, and then based on this estimated model, directly characterize the distinctness of the patterns. So this is again the classification situation. And now instead of classification, what we do is we fit multivariate normal distributions to the data. Each normal distribution is characterized by a centroid, which is shown by a cross, and variance covariance error matrix, which is indicated by the contour lines of the normal distribution. They are of elliptical shape for multivariate normal distributions, or ellipsoids in higher dimensions. And the highlighted contour line corresponds to something like 1 standard deviation of this normal distribution in high dimensional space. And then we can look at the distance of the centroids in this voxel activation space, and we can measure it in units of the error standard deviation, which is here indicated by these ellipses. And for example, in this situation, we get something like 1.5. This measure is also called Mahalanobis distance. It's a distance between centroids measured in units of some kind of variance, which here is the error variance of the data. You can see that it's 1.5 because the distance from the centroid to the contour line is 1, and then it's just half of that again until the next centroid. That is kind of a multivariate version of the well-known effect size measure, Cohen's d, which is the same thing in 1 dimension. And then it doesn't work that well anymore if we look at more than 2 classes, because then you have 3 different distances. There's a question of how to integrate those. So instead of distances, what is more generalizable is to look at variance. Now here's a white ellipse indicates the variance covariance matrix of the 3 centroids. So how much variance is there between these 3 points? And this is compared to the error variance covariance, which is indicated by the colored ellipses. And that is finally the measure of multivariate effect strength. Does this make sense so far, or are there any questions?
Excellent. Perfect. Yeah.
Mm-hmm. Now, that's a starting point for cross-validated MANOVA. Now, the nice thing about this approach, looking at multivariate normal distributions, looking at explained variance, etc., is that it can be done using the same generative model that we use for univariate fMRI analysis, the general linear model. I call it here the multivariate general linear model to emphasize the multivariate character, so we have more than one dependent variable, but actually the GLM is from the start a multivariate model. This is the equation for the general linear model. Y equals x times beta plus xi. Y is the fMRI data. It's a matrix with n rows and p columns. The n rows correspond to scans or volumes of fMRI. The p columns correspond to p different voxels. Now, these data are described by a design matrix made up of regressors. The design matrix has the same vertical dimension as the data. It also consists of n volumes or scans, and it comprises q different regressors. And then there's the model parameter matrix, beta, which correspondingly has q rows and p columns. The q rows correspond to the q regressors, and the p columns correspond to the p voxels. That means for each regressor and for each voxel, there is one number, one model parameter, and that is arranged in this matrix beta. And then the design matrix is not sufficient usually to describe the data Y completely, which is why there's a residual, an error. And this error has the same size as the data matrix Y. It consists of n volumes or scans as rows and p columns or voxels. About the error variance, we make the assumption that each row in this matrix comes from a multivariate normal distribution with mean 0 and a variance covariance matrix, which is called sigma. And this sigma matrix is of size p times p, so it is voxels times voxels. Now, in standard univariate analysis, one would use this same model, but one would look at the parameters for each voxel separately. And what we're going to do is to look at the parameters for the different voxels together to have a multivariate analysis. This is a visualization of the general linear model for multiple voxels. So on the most left-hand side, you can see the data matrix. The gray shades correspond to values of the bold signal measured in a scanner. The signal is measured in 10 different voxels over, I don't know exactly, something like 100 scans. It's modeled by a design matrix, which consists of 4 condition regressors and 1 constant regressor, number 5. There are a number of events, 2 events, experimental events for each condition. They are convolved with the hemodynamic response function, which is why you have this appearance of like a ramp or a knob. And this is combined with a parameter matrix, beta. It has entries for the 5 regressors, rows, and entries for the 10 voxels, columns. And this is simulated data, obviously, to make it nice to look at. And you can see that in condition 1 and condition 3, there's not much going on. The parameter values are close to 0, close to black. But in 2 and 3, there's a distinct pattern. And if you then look at the time points, the scans, where the corresponding condition is active in the design matrix, you can see that a similar pattern appears across the voxels in the data. And finally, there's error matrix xi over scans and voxels. Now, what do we do with this? The same way as we do in univariate analysis, we define an effect of interest by a contrast matrix. Contrast matrix, for example, has a +1 for one condition, a -1 for another condition, zeros for those conditions that are not of interest at the moment. And then this contrast matrix can be used to extract from the parameters that part which belongs, which corresponds to this effect of interest, which is defined by the contrast matrix. Technically, that is done by multiplying the parameter matrix beta by the pseudo-inverse with the contrast matrix, and then multiplying it with the contrast matrix again. These two things together, C pseudo-inverse, that's the so-called projection matrix, meaning it takes something and produces something of the same kind. So delta is still a parameter matrix, but it eliminates everything which is not of interest and retains only what is of interest. For example, a difference between conditions 1 and 2. And using this contrast, we can now decompose the GLM, the model, into 3 parts. We can now write y equals x times delta. That's the part we are interested in, the design matrix multiplied with those parts of the parameters that correspond to the effect we're currently interested in, plus x beta 0, and beta 0 is beta minus delta, so it's exactly the rest which does not fit into delta, the so-called reduced model, plus psi. So this equation is equivalent to the equation I showed before, y equals x beta plus psi, but now we have split beta into 2 parts, delta and beta 0, where delta corresponds to the contrast and beta 0 contains everything else. So now we have a description of the data in terms of an effect, the rest, which is not the effect we're interested in, and the error. And in cross-validated MANOVA, this is the measure we use, which are called pattern distinctness. And it is based on the delta part of the parameters, the part of the parameters singled out by the contrast, and the error variance. You will see there's x delta in there. So that's a part of the data, in a way, the reconstructed part of the data, which fits in with the effect we're interested in. And then it's multiplied by itself, but transposed. These prime characters are supposed to indicate matrix transposition the same way as it is done in MATLAB, for example. The two are multiplied together. And because x delta is an n times p matrix, so scans times voxels, and it is multiplied with its transpose from the right side, what we get out of this is a p times p matrix. It's a p times p matrix, which is a variance covariance matrix, which is a part of the total variance of the data, which corresponds to the effect we're interested in, where this effect is defined by the contrast matrix C. And the next term is based on the error variance, and we invert it. So effectively, we divide the effect variance by the error variance. That is still a matrix, which is p times p, voxels times voxels. And finally, we apply the trace operator to this matrix. The trace operator is nothing but the sum of the diagonal elements. This is a definition of pattern distinctness, and it quantifies the ratio of effect to error variance in the multivariate setting. So this is because we have the separation between different parts of the variance, the effect variance, the rest of the variance which can be modeled, and the part which cannot be modeled, which is the error variance. It's a kind of analysis of variance, but we do it in a multivariate way. Therefore, it's a kind of multivariate analysis of variance. So this white ellipse basically represents the first term under the trace, delta transpose x delta, which is a variance covariance matrix of the part of the variance that belongs to the effect. And all of the colored ellipses have the same shape. They all correspond to the error variance covariance matrix sigma. And I said before that we're going to compare them. So we compare the white ellipse to the colored ellipses. And concretely, we do that by multiplying one with the inverse of the other. And then we still have a p times p matrix, which is inconvenient. We want a single measure of multivariate effect strength. And that's why we use a trace. There are possibilities to do it differently. One could use a determinant. There are a number of variations. I chose this version because it's closest to this idea of a Mahalanobis distance. So in the case where we're just comparing two different conditions, this d can be interpreted as a squared distance between the centroids, measured in terms of error standard deviation. Okay, so this is a theoretical quantity. All of these, we could compute this if we knew exactly what is this delta part, but in practice we don't know it. We can only estimate it from the data, and that's where the cross-validation part comes in. So how do we estimate this? First, we estimate the parameters of the GLM. Estimates in the following are indicated by the small hat on top of a variable. So beta hat, those are the estimated parameters, and they're estimated from the data in the same way as it is always done for a linear model by using the pseudo-inverse of the design matrix X. Then we estimate the errors as the data minus the part that we could model by the design matrix, y minus x times beta delta. X times beta delta is the part that can be modeled. Then we estimate the contrast, or rather the parameter difference matrix, if you will, the delta matrix. Delta hat is C pseudo inverse beta hat. That's the same formula as for the theoretical quantity, only that now we use the estimated parameters and therefore get the estimated parameters corresponding to the effect. The error covariance, variance covariance matrix is estimated from the residuals psi hat, again by multiplying with the transpose and dividing by the residual degrees of freedom. And now the simplest thing we could do is to simply plug all this into the formula that I showed on the previous slide. So instead of delta, we have delta hat. Instead of sigma, we have sigma hat. That is what is in the last line on the slide. And that is indeed something that you can do. It's called the Bartlett-Lawley-Hotelling trace in traditional ways to do MANOVA. And you can use this as a test statistic. The problem with this is that you can't really use it as a measure of multivariate effect size. And the reason for that is that there's a square inside. So we multiply x delta hat with the transpose of x delta hat. We multiply something with itself. So there's a nonlinearity in there, a square in there. And the problem is if we have an error in there and we square, then in the square, the error can only go in one direction. So delta hat, it may be a little bit above the true delta or a little bit below the true delta, but x delta hat multiplied with itself, it can only be above the x delta multiplied with itself. So we have a bias in this estimator, and that's why Bartlett-Lawley-Hotelling trace can be used as a test statistic, but we don't want to test at this stage. What we want is we want to quantify a multivariate effect strengths, and that is why we can't use that. Now, how do we get around this bias that comes out of multiplying a noisy estimate with itself? Cross-validation. Now, this is a little bit unusual use of cross-validation because the usual context in which cross-validation is used is that you train a classifier, for example, so you train an algorithm, and then on the test data, you see how well does it work. You use it to quantify what machine learning people call generalization error. What is trained here, there is not actually anything trained here. There's no algorithm. We have estimated a model. So we are not really interested in the performance of an algorithm. We're interested in estimating a certain aspect of this model. So train and test has to be taken with caution. They're just here for analogy with a procedure when using classifiers, but it's not really training and not really testing what happens here. What happens here is that we separate one of the two delta hat, the one on the left, furthest on the left, before multiplying it with the other one on the right. And we estimate the delta hat on the right side from data which is independent of the data which is used to estimate the other delta hat on the left side. So delta hat appearing twice here in this formula does not denote the same quantity. It denotes the same kind of quantity. So the parameters corresponding to an effect defined by a contrast in two independent pieces of data. For example, one run of fMRI and another run of fMRI. And because now these different parts are statistically independent from each other, we don't have this problem of errors only going in one direction anymore. That can, if you're interested, I can show you the proof, but for the moment, just believe me that when we do it this way, we get an unbiased estimator. The formula, the first formula provides an unbiased estimator of the formula, the second formula, which is a definition of pattern distinctness. And this is now for one piece of train and one piece of test, but we can do this with different train and test data sets, average across it, and after averaging, it's still an unbiased estimator. So this is the idea of cross-validated MANOVA. We do the multivariate GLM, we decompose the variance, which is the ANOVA part, and then we use cross-validation to get an unbiased estimate of the multivariate effect strengths as defined by pattern distinctness. Let me see. Yeah. Okay, so this is what we did in 2014, and I think it has a number of advantages over classifier-based MVPA. So we can use DES, we can use classifiers to look at the distinctness of patterns for two classes of trials. But because it's based on a decomposition of variance, we can also use it for more than two classes. We can even use it for something like a main effect and interaction in a factorial design, which is hard to do, if not maybe even impossible with classifiers. And it can even be used without classes. So for example, if you have a parametric regressor where the contrast basically says pick out this single regressor and do not compare to anything else but itself, or but zero. This is a publication, Allefeld Haynes, 2014. This is a list of plenty of plenty. I'm happy about this list of publications which have used it. You might notice at some point there is Schmidt and Blankenburg in there, top of the second column, but there are also several publications together with Thomas Christophel, who has been one of the most enthusiastic users of cross-validated MANOVA. And this concludes the first part of what I want to say, and I would now turn over to Thomas to give a concrete example of cross-validated MANOVA.
Yeah, so if this is the Super Bowl, this is the halftime show, basically. And yeah, as Carsten already said, like we've been kind of the most joyous users of CV MANOVA because it's so flexible, because it allows a lot of things, right? And Carsten here talked about the application where all of this started with was like classification examples. But the one thing that we kind of learned really relatively quickly, thanks to Carsten, is that you can use this for much more than classification. And this is an example for model comparison. And I'm just going to give you a theoretical intro because I can't help myself doing that, because this is about color categories, right? And I'm a work memory dude, so I'm always interested in what do people do when they memorize stuff. So if you memorize a color like this, you might, in some way kind of represent the particular color in as much detail, probably not numerically like this, but in some kind of very precise, these are exactly the color values that I see. Or you could just say red. Yeah, so you could categorize the stimulus. And we know already from behavioral data that people sort of do probably both to some extent, right? And now this is a study done by Chang Yan, and it just came out in NeuroImage if you want to read up on it. And this is a very, very simple working memory task. So what you see is, in the end, what you see is two colored samples, and they take this particular shape, but that's not really relevant here. It's just one color and another color. And then there's a cue telling you which of these two to remember, right? In this way, we control for perception so that you have this stimulus, the second stimulus is the one that you memorize in working memory, and the other one is basically a thing that you can forget about after you've seen this cue. Then there's this really long delay, and that's what we're interested in, and then subjects actually have to recall the color. So if people are memorizing this color, they actually try to reproduce this on this color wheel as precisely as possible. Now, we want to know whether people use colors, color categories in this task and how they are represented in the neural data. And for that, we actually use two separate tasks that we do after the scanner. So after the scanner, people have been doing this task for, in this case, for 4 sessions, right? So like something like 6 hours of fMRI. Yeah. And then they we get them back to the lab and ask them to do these very simple tasks where you see a color and are asked to pick a label for it. This is what we call category naming. And this will give us category boundaries. So for example, if you see here in the middle, this is green. And these would be the boundaries for different subjects. The subject and the little white circles, right? So there is some variation across subject, but more or less, there are shared boundaries between this. And another thing that we can do is actually ask them, hey, what actually is, for example, pink, right? And so here they are queued with a name, and they tell us where is that color on our color wheel. And this gives you the kind of the prototypical color, right? The most green, the most blue, the most purple, and so on. Right? And this is very much based on the behavioral data that I mentioned already before, where they found out that behaviorally you can use data like this to predict the response in a color working memory task like this. You know? And we just basically took this behavioral task and kind of bastardized it to create an encoding model, right? Because this is actually the behavioral data for this, right? So here you actually see that subjects actually have these color report bias, but I'm just going to skip this. Yeah? So this encoding model we just basically do by averaging these two naming frequencies. So basically you take here the naming distribution and the identification distribution and get this regressor. And this regressor is a thing that we're going to fit into our multivariate GLM to see whether it actually explains the data well. Yeah. And basically the way you can think about this is this would predict the response of a categorically tuned neuron or voxel in the brain that cares about green. So, yeah. So for the most green stimuli, it would respond strongest, and that response would go down all the way till stimuli turn more bluish, and then kind of the probability goes down. And what this is it's a graded categorization model. A stimulus isn't always categorized in the same way, which is kind of counterintuitive, but if you look at the neural data, for example, also in electrophysiology, that is what you see for categorization. A categorization is never really all or none. Yeah? And we kind of took this empirically based categorical model to a sensory encoding model, a model that basically assumes that colors are similar to each other, but the similarity is basically just governed by the distance in our use space here. Yeah? So no categorization, no prior assumptions about where blue ends and where green ends and so on. And you can basically take this model like this, or you can shift it around as much as you like. The results are very much the same here. Yeah? Okay, now here's where the CV-MANOVA comes into play, because this is now a thing that you cannot do anymore with conventional classification. First of all, we have some kind of areas, right? This is visual cortex, for example, primary visual cortex. And we have lots of trials for different colors, and we have lots of voxels. And we filter this voxel-wise data through our encoding model, right? So here, I think in this task, we had 50 different colors. And then basically take this data through our encoding model. And what you get is then not voxel responses for individual colors, but voxel responses for a little individual color model parameters. So basically for the orange model, for the yellow model, for the green model, blue, until the pink model here in the end. Yeah? And this is we do this by basically taking these model parameters and fitting it to a GLM, right? So these are already parameter estimates in Carsten's explanation, right? So that you can basically take our encoding model times the parameters and get back to the original data. Now, there is a technique called inverted encoding models that have been using encoding models like this in the past. And what they would do at this point now, invert the encoding model. So turn this model into not a prediction of data, a tool to explain the underlying data, but a tool to predict the color in a given trial. That has been a really prolific way of doing analysis like this. But the limitation here is that you need to have something that you want to predict, that you think your model actually is good at predicting, right? So in a very extreme sense, for example, if you have kind of not a graded, but a kind of boxcar-like categorical model, where all greens are exactly the same, that model might be a true descriptor of the neural data, but might be terrible at predicting the behavior or the incoming stimulus, right? Because in this model, all greens, for example, are the same, and that means that you cannot use it to predict different greens against each other, right? So that's why inverting here the model would be wrong, because you basically make your model worse. Instead, we're using cvMANOVA, and because basically what cvMANOVA does is it predicts the data in the left.run. So right, so we train these parameters in, I think in this case, I think 16 color working memory runs. So we train it on 15 runs and test on the 17th, right? And then you have this cross-validation step that Carsten told you around, and I think this is just another way of kind of visualizing this, right? So if, for example, here you've got 4 runs, and each of these arrows represent a difference between 2 channels, so say 1 and 2. In these 4 runs, you would basically train on 3 of them, say the lower 3 here, and see whether the direction of the arrow is the same. So whether the difference between these 2 parameters is the same in the training and the test set, and basically if the difference, the direction of the difference is the same, then you have a positive D, and if it's the opposite, you have a negative D, and if there's just no reliable relationship, you basically get D more or less 0. And so basically all this is asking is basically, is there a consistent difference between these color channel activations? And then you end up with pattern distinctness D, and you can do this for our categorical model, our sensory model, in the working memory task, and we also do this for another task where we don't have work memory, where people are just seeing the stimulus and reporting it immediately, so that we have a kind of perceptual baseline for this. Okay, so this is the results. 3 areas. One is primary visual cortex that probably has color information, but is not kind of known as the color area. And here and then V4, V1 are typical ventral stream color areas that we see in the literature. And basically here, this is the comparison between the sensory model and the categorical model in primary visual cortex, and we basically see no difference here. And what's important to know here is that because these 2 models are correlated, they both describe the same properties, color, right? They can have, if one is the true model, or one is more similar to the true model, the other one is still expected to have information, right? So you can only really interpret this if there is a significant difference between these 2 models. So here you see that basically, yeah, there is clearly information about color here, but we cannot tell what nature it is. But if you go a little bit deeper, you actually see that in V4, V1, during working memory, these representations become more categorical during working memory. So here in V4, actually the sensory model only loads to a not significant level, right? I mean, more importantly, there's a significant difference between the sensory and the categorical model, meaning that apparently the underlying data in V4 in the working memory delay shares more features with the categorical model than the sensory model, right? So, and this is the important part here when you're talking about model comparisons. These are just 2 possible models of, millions of possible models that you can think of. And so we cannot exactly say, okay, the data looks like the categorical model, right? But we can just say, well, the data is probably more categorical than sensory in this comparison. And the interesting part is that this effect is actually limited to memory. So if you have an undelayed task where basically you see the stimulus and you report it immediately while you're seeing it, we don't see this effect. And even so that here in VO1, we actually have a significant interaction effect between the 2, meaning that the information present in the working memory delay is significantly more categorical than during the perceptual task. And yeah, that's why cvMANOVA is a very, very critical tool for our work, because again, you couldn't do this with a classifier because classifiers basically rely on something to classify, right? And in the end, what you would be classifying is here still color, right? So there wouldn't be much of a difference here. No? Instead, we can basically say which encoding model explains the underlying data better, and that's why I love it. And we'll have more about this in the second example, but I think Carsten first needs to explain how that one works.
Okay, cross-validated cross-MANOVA. Now, there's a variation of classifier-based MVPA where you don't just train on two classes of trials or on two conditions and then test on the same ones in another part of the data, but you train on another pair of conditions. So you have four conditions, for example, A, B, C, D. They are here symbolized by the colors green and magenta and blue and orange. So you create a classifier for one pair of conditions or one pair of classes, and then you test this classifier on the other pair of conditions. I have here shown two different classification boundaries. So the green magenta one would come from the green and magenta data points. The blue and orange one would come from the blue and orange data points. But cross-decoding means that you use the green magenta classifier in order to classify the blue and orange data points. The idea here is that you can have a manipulation, or you can have pairs of conditions which where the difference between the conditions is in some way similar, but there's something else that has changed between them. One example would be distinction between two different objects, and in one case they're actually present on screen, and in the other case they are remembered or imagined or something like that. And then the idea of cross-decoding is that you can see how much the representations, as far as we can access them through fMRI, are more or less invariant. Now, the question is, how do we deal with this in this MANOVA framework? It's not straightforward because, well, it's not just about different explained variants anymore. As it will turn out, it's more about explained covariance. Let's see, if we go into the modeling view, then we would have four different multivariate normal distributions. Each of them has their own centroid, indicated by the cross, and we still assume that the error covariance, variance covariance matrix is the same across everything, indicated by the colored ellipses. Before I showed you the distance between the centroids without an orientation, now I have attached arrowheads, so you can see that what the comparison is like. So the question here is the representation that is present in the difference between green and magenta the same as the representation that is present in the difference between blue and orange? And if we look at it in this modeling view, where we look at differences of centroids, directed differences of centroids, therefore vectors, in this specific situation depicted here, we would say yes. This is the exact same representation. So there's some other stuff going on. The difference between green and blue, which is the same as the difference between magenta and orange, is due to some other things that distinguish these conditions. For example, actually visually present versus imagined. But the difference between the two classes, which are somehow analogous, so green and blue would be analogous, magenta and orange would be analogous. The respective difference between them in their pair is exactly the same. Now, here it's interesting to note that the Mayer-Nobis distance I talked about, which was sort of generalized to multivariate variance covariance, is, as all distances are in mathematics and linear algebra, based on an inner product. So basically, when you look at the distance between the green centroid and the magenta centroid, you're looking at this vector between them, and then you're multiplying this vector with itself. That's how you define a distance, or to be precise, a squared distance, and then you can apply a square root to make it a proper distance. And because of that, we can now make the inner product between two different vectors. So we multiply the vector from green to magenta with the vector from blue to orange. And if they are perfectly aligned and they have the same length, then we will get the same result, dx equals 1.5. The Mahalanobis distance in the first pair is 1.5, the Mahalanobis distance in the second pair is 1.5. But if we multiply them, because they're exactly oriented in the same way, the inner product is also 1.5. Now, what happens if we move one of the condition centroids around? So, for example, the orange centroid is a little bit lower along the voxel 2 axis. Now, the two arrows are no longer aligned. They still have roughly the same length, but they're no longer perfectly aligned with each other, and therefore the inner product becomes smaller, in this case 1.2. And we can go further until this situation where the two vectors are oriented in such a way that the inner product is 0. You will see that in the picture they're not perfectly perpendicular to each other. That's because this inner product is calculated with respect to the error covariance the same way as the Mahalanobis distance was calculated with respect to the error covariance. You can imagine it if you were to squeeze this diagram in such a way that the ellipses, the error ellipses, become circles. Then after this transformation, you would see that the two arrows are actually perpendicular to each other, and that is why this inner product is 0. Okay, and before we went from the Mahalanobis distance to variance covariance corresponding to an effect, and now we're going to, well, covariance, if you will, corresponding to an effect. Let's first look at the second formula. That is the definition for pattern stability, which is a variation of pattern distinctness for two different contrasts, CA and CB. So on the one side, for the effect covariance now, we have X delta beta. Delta beta is the same as before, except where it corresponds to the contrast B. X delta B, this corresponds to the contrast CB. So this works in exactly the same way as before. We have a contrast, we apply it to the parameters, and we extract the part of the parameters which corresponds to the effect encoded in the contrast CB. On the other side, we still have the transposed part, X transposed, delta transposed, but now the delta is BA. There's a second contrast, CA, and at first you might imagine that we simply have delta A here, delta A transposed. This doesn't work because the contrast CA, which is for, to stay in the picture from before, is a contrast which involves the two other regressors, because they don't fit. Because in this case we would go for regressors with events which happen at a different time than those regressors which are involved in contrast B. Because of that, we have to kind of mix the contrast. So delta BA is defined as CB times CA pseudo-inverse times beta. So the first part is the same. We multiply beta with CA pseudo-inverse. That kind of extracts the part that corresponds to the contrast A. And then we would usually multiply it with CA to go back to the full parameter matrix, which is reduced to only the effect that is encoded in CA. But we don't, we use CB. And the reason for that is that then regresses match up, and this inner product, which defines the effect covariance, actually works. Okay, so before I always talk about variance covariance matrices. A variance covariance matrix is, if you will, the multivariate analog of a variance. If you have univariate variables, you compute the variance between them, you have the variance. If you have multivariate variables and you compute the variance, what you get is a variance covariance matrix. But this whole variance covariance matrix is a multivariate version of a variance because it describes the relations between a set of variables and themselves. Now, in the modified version, the term marked effect covariance, we're looking at the relation between one set of variables and another set of variables. So it's again a matrix which is made up of covariances, but now this whole thing, this whole matrix is the multivariate analog of a covariance. So what we're looking at now is no longer the relation between effect variance to error variance, but we're looking at the relationship between, or the ratio between effect covariance and error variance. But otherwise, the logic is the same. We multiply one with the inverse of the other and take the trace. So this is cross multivariate analysis of variance, or cross-MANOVA. Now we can combine that with the strategy used before, the cross-validation for estimation, and it's pretty straightforward now. So we estimate the delta BA from trained data, and we estimate the delta B from test data, and multiply them together. And yeah, and that's the estimator of pattern stability. You will notice that if you make CA and CB the same thing, so CA equals C and CB equals C, then this whole formula reduces to pattern distinctness. So pattern distinctness is a special case of pattern stability for the case where the two contrasts are the same. Okay, and putting in the cross-validation step as the estimation strategy to get unbiased estimates, we arrive at the full thing, which is cross-validated cross-MANOVA. And this is something we have already used, triggered by the interest by Polina Jamshchina in her PhD. We had one paper together about that, and then there's another publication by her and Thomas and other people who also used that, but we have never properly described that. So this was like a paragraph and message section. And now what we're trying to do right now is to do this properly. So we want to write this up as a new paper. And then we haven't published the code for this cross-analysis before. So there will be a new version of the CV MANOVA toolbox to implement this method. And that's my part. And now, okay, are there any questions before Thomas shows an example?
I only know how to unmute myself when I'm not sharing the screen, so I had to quickly unshare it. Yeah, so I made Carsten do this because, you know, as you probably already noticed, I've been falling in love with CV MANOVA like for a long time, and we basically want to use it for everything. And there are applications where we want to do cross classification. And actually, the first time we used it was not doing Paulina's PhD, but doing a master thesis. So that says a lot about Paulina already, that she kind of stormed herself so much into this stuff that she needed all of these new tools. Now, the example that I have right now is one that is a little bit simpler than the color one that you just had. So I'm going to quickly introduce this. And this is again about working memory because I don't do any other research, sorry. But basically, one of the stimuli that we probably use most often in working memory is some kind of Garbo patch orientation. Yeah? And you know, when you see the stimulus, it's a symmetric stimulus, right? So it's actually mirrored across this point. When you see something on the left up here, it's also down here on the right. Yeah? Now, the funky part is if you actually ask subjects how they memorize it, and particularly how they would label these internally, right? We talked about color labels earlier. Now, these are labels for orientations. They actually say that orientations up here that are kind of rotated counterclockwise from the vertical are left stimuli. And the ones that are rotated clockwise from the vertical are called right stimuli to most of the subjects. So this is naming data, and this is actually in the setup where they are freely naming these kind of stimuli. Some of them also call them weird pies and stuff like that. So they use all the silly names, but quite frequently they just call them left and right, you know? Based on whether they are rotated left and right on the upper part of the screen. So which is basically telling you, hey, these subjects clearly care more about the upper side of the stimulus than the lower side. You know? And we thought, okay, well, if this is the thing when they are asked to name them, is it actually part of the internal representation? No? And for that, Joanna Seabra, who is a PhD student in the lab right now, actually have done this rather massive acquisition of data of both orientation and location working memory. Yeah? So these are the two kinds of trials that subjects have, either orientation data with kind of orientations from all over the orientation space. There is a long delay, a very long delay of about 15 seconds. And then they are asked to rotate the stimulus to match the original orientation. And the other type of trial is just the same with location. So you have locations that can be on this imaginary circle around the fixation point. There is again a long delay, and after the delay, you're asked to rotate this dot to the right position that you memorized. And again, what's important here is that the delay is extremely long, so we can actually see how the signal kind of unfolds from the original encoding of the stimulus all the way towards the end of the delay. By the way, this is actually scanned kind of if you kind of, from my point of view, into your room, you look to the right, so down the hall, that's where we acquired this data at the CCNB. Great scanner, great multiband sequences. Thank you for that. And before I show you how we analyze this data, just a quick primer, because we already have quite some insight in how these items are represented in working memory. And this is kind of your standard areas of the dorsal stream that you would look for both orientations and locations. And that already suggests that both orientations and locations share a neural representation. And the assumption behind this is that they both use some kind of spatial coding scheme. Yeah? Now, for the analysis, there is this peculiar thing about orientations that if you have them kind of for these Gauss patches, they actually describe a 180-degree orientation space, because if you take this orientation and rotate it and rotate it and rotate it until it's here, then you actually end up with the same orientation again, right? So you have an orientation space of 180 degrees because they're symmetric, whereas locations have a 360-degree orientation space. So the question is, if you want to ask, hey, how do these two representations share a representation space? You actually have to make assumptions how these map onto each other, right? And the first assumption that you can make is that if there is an orientation that goes from the top left to the bottom right, or a left orientation if you ask our subjects, then they're probably represented by a spatial code here and or there. Now, from this naming data that we saw, we actually could assume that, hey, people care more about the upper part, so maybe actually the upper part of the spatial code is more important than the lower. And that's exactly what we're doing. So we are training a cross-MANOVA model to distinguish these upper locations and test it on the orientation space. And we can do the same for the lower part of the space. And that then results in this kind of plot. So this is visual cortex, the areas V1 to V3. Here in the beginning, you see the onset of the samples, stimulus. Here is the recall. So these are these 15 plus seconds in between. Was there a question? No, okay. All right. And what you see is that, first of all, in the beginning of the delay, you don't see much information about the stimuli at all, because the hemodynamic delay. And then when information kicks in, you actually see in the beginning that both the upper and the lower location model is really good at predicting the orientations. But then already very early in the delay, you see actually that the upper location model wins out and wins throughout the whole delay. So this already kind of shows that, okay, there is a change in the neural code for working memory across this delay. Now, if we're looking at different areas, and this is kind of looking from posterior to anterior, so this is V3AB, kind of like in between visual cortex and parietal cortex. This is IPS, and this is FEF. You see that in visual cortex, actually the same pattern of results holds true. So in the beginning, both models are equally good at decoding the, at explaining the variance in the orientation task. I'm always have to remind myself. Then if you look at these more anterior regions, IPS and the frontal eye fields, actually the upper model wins out completely, very much from the beginning, such that even in FEF, the lower location model actually does not explain any variance in the orientation tasks. And so this way we have actually a direct access to understand how people internally represent orientations in working memory. And I think this is a really cool tool that we can use to understand neural codes by kind of generalizing between stimuli. Now, what I want to quickly add here is that this analysis is probably also possible in some kind of classification way, say with support vector machines. But the reason why we're using it here and the reason why cross-MANOVA is such a crucial tool is because on top of this, we of course want to put encoding models, just we didn't for the color in the previous example, so that we can basically combine the goods of all of this. And I think that's the thing that always makes me excited. But maybe just to give you one last example, how general you can do this, and also, by the way, how fast you can actually run this, because the newest implementation is very, very fast. And that is, you can actually generalize this, the upper and lower analysis to all possible rotations of this location model, right? So the original model that we have kind of was training on these upper locations, but you can also train on these kind of locations, right? And so what you quickly see is that you run into models that actually do not make sense, right? You're assuming that basically orientations are coded by an orthogonal location, right? So if this is the orientation, then the location here is predictive of that, huh? And this is exactly also what you see, right? So this is basically a different kind of representation of this data, right? And so here at 0, there is the upper model. At 180 degrees, there's the lower model that we just saw in the previous plot. And this is in the early delay in our 4 areas that we looked at. And you see that here in visual cortex, the upper model wins out, but the lower model is also pretty good early in the delay. Later in the delay, actually, the upper model wins out way more. And all other models are just worse, right? So this is kind of this elongated shape that you see here and there as well. And then, so you can see, okay, there's information in the upper and lower model, but then here, the more it turns into memory, you have this stronger and stronger right tilt in the predictions, meaning that the representation is more asymmetric. The upper model is better than the lower model. And, this is just a kind of different way of looking at this data, and kind of gives you a hint at what kind of different analysis you can do with this cross-MANOVA. And so that's basically all I have. And I think we're already running deep out of time, but maybe there are some questions.